Okay, today I'm not going to present informations about optogenetics, but I will present a very closed field, and that is a bioluminescence science. I mean that they use some light information in the diagnosis or research, and the central part is the Luciferase. Luciferase is an enzyme from the flies, and it's a light-producing enzyme naturally found in insects, fireflies, and in luminous marine and terrestrial microorganisms. So, when in the eastern part of America, in the summer night, on the grass, we can find a lot of fireflies. It's very beautiful. And it also in Detroit, when walking around the campus and during the sunset, after sunset, there's a firefly that I can see. That's just amazing. And this small creature and have this can illuminate can emission light. This property is due to this enzyme luciferase, and this image is from PDB protein database. And the protein part is a luciferase, and it has a cofactor that's a D-luciferin. And a chemical reaction is that after the luciferase binding this cofactor luciferin, then it uses the energy from the ATP and it required the oxygen, and it exists of the magnesium, and it will hydrolyze the ATP to AMP and emits CO2 and a photon. So luciferase is a protein part, and the cofactor is a chemical molecule, luciferin, and it required energy is from the ATP. And since it's changed oxygen into CO2, so it's a kind of like, and when it's breathing, it will at the same time emission some light. So we can see that at the night, the fireflies emission the light is not a continuous light. It's the shining, then dark, then bright, then dim. And this process is a quite energy, how to say, it's an energy consuming part because emit one photon, it requires 8 ATP. So this consuming a lot of energy. So we can say that so such amount of energy required, we know that there's a lot of ATP there. So this property is being used on the cancer research, because on the cancer cell, there's a very high metabolic reactions going on at the cancer cells comparing to the normal tissues. So after injection the mice with the antibody that was binding with this luciferase enzyme, then also inject the cofactor, those luciferin, and do the imaging, then we can know that where is the cancer located. And this is a kind of a non-invasive procedure, and Of course, this is so far is the animal research. Because people, I don't think it's a good idea. I will check that later, but I don't think it's not a good idea to emission some light from the human body. But there's a advance is that because this is injection is an antibody, so it can be removed by the human body's immune system, so it's transient. It's not stay there all the time. Yes. So thank you. And since this contribution of the firefly to our human beings' health and happiness, then please appreciate its contribution and save the life of fireflies. Thank you.